download our Revise It Right revision app for hundreds of videos, quizzes, exam questions, tutor support and so much more. This video is about drugs. Now some drugs relieve symptoms and others actually provide a cure. So we'll all be very aware of the drugs painkillers. Now the job of painkillers is to relieve pain. They don't actually tackle the cause of the disease, whatever it is that you've got, or kill the pathogens. Their job is just to reduce the symptoms and therefore to relieve pain. Other drugs, however, actually do reduce the symptoms by tackling the underlying cause. So for example, antibiotics, they actually kill or prevent the growth of bacteria that are causing disease. So for example, in previous videos we've looked at salmonella and gonorrhea as being examples of diseases caused by bacteria. We can use antibiotics to kill these bacteria or to stop them from dividing and growing to really deal with the problem, to try and cure. Okay, so that's what we mean when we say some drugs just relieve, i.e. painkillers, and other drugs actually provide a cure, i.e. antibiotics. Now it's really important to understand that antibiotics can only be used on bacterial diseases. Okay, and under no circumstances should they be used for a viral disease. And that really is because they cannot kill or destroy viruses. You'll remember that viruses reproduce using your own body cells, which actually makes them very difficult to treat. And antibiotics cannot treat viruses. So when we looked at HIV, when we looked at measles, these cannot be treated with antibiotics, but salmonella, gonorrhea, they can, because they are types of bacterial diseases. Now many drugs originally came from plants, okay, and we're going to look at some example of these in a second, but plants produce a variety of chemicals that defend themselves against pests and pathogens. Now some of these chemicals we can use to our advantage, and we can extract them from the plant and insert them and place them into drugs to treat human diseases or to relieve symptoms. And a lot of our current medicines have come from plants. So for example, aspirin. Okay, now you may or may not have heard of the drug aspirin before, but it's actually a painkiller. Okay. And it also lowers the level of fever. And it was developed from a chemical found on the bark of a willow tree. So here is one example really of how we're using these chemicals produced by plants to our advantage. A second one that we need to learn about is Digitalis. Now you may or may not have heard of Digitalis, but it is used to treat heart conditions. Now this was actually developed from a chemical found in foxgloves. Okay, so you do need to remember that aspirin, which is a painkiller, originally it came from a chemical found in the bark of a willow tree, and that Digitalis, which is used to treat heart conditions, was found in foxgloves, the plant foxgloves, and we obviously use it to treat heart conditions. Now, not only do we find and discover drugs from plants, but we also extract some drugs from microorganisms, and the main one, and a really big example, is penicillin. Okay, now penicillin was discovered by a scientist with the name of Alexander Fleming. Now you may or may not remember the story around Alexander Fleming, but the story goes that he was growing bacteria in his labs, and then he went on holiday. And when he came back, he realised there was loads of mould on one of his dishes, on one of his Petri dishes. And what he noticed around that mould is that it was free from bacteria, i.e. there was no bacteria around that mould region. He therefore found that this mould on the Petri dish was producing something, a substance that killed the bacteria. And that substance that was being produced was called penicillin. Okay, so he had actually found this chemical penicillin that could kill and destroy bacteria. Okay, now that is massively used in antibiotics to kill and destroy bacteria, really for many diseases now, and it's really been used to our advantage, and that was accidentally discovered by Alexander Fleming from mould. Nowadays, drugs are made on really large scales in the something we call the pharmaceutical industry. And really this is when we're synthesizing different drugs in a lab by sort of mixing different chemicals together. However, this process may start with a chemical extracted from a plant, or it may not. But really, the pharmaceutical industry is a massive billion-dollar industry now, and that is where most of our drugs now get developed. Remember to download our Revise It Right revision app, watch over 700 videos, answer 4,000 plus quiz questions, over 1,000 flashcards, 1,000 exam questions, worksheets, forums, and get help from qualified teachers and so much more. The link is in the description. Now, drug testing is a very long process, and really what that process involves is the discovery of the drug, i.e. maybe the synthesizing of that drug in the lab, through to all the stages that get that drug on sale for humans and us to buy to cure or relieve our particular disease. And that process is very long, and it involves numerous amounts of stages. So obviously stage 1 is that discovery, i.e. a drug is being discovered and developed. Now once a drug has been discovered and developed, it needs to go through loads of stages of drug testing. Okay, and the first stage of drug testing is preclinical trials. So preclinical trials. Now preclinical trials are before humans. So these trials will be on animals and on cells prior to them being used on humans. Okay, so in the first stage of preclinical trials, it's tested on cells and tissues. Okay, now really in this particular stage and in drug staging in general, we're looking for three things. We're looking for three main things. The first is something we call efficacy. Now this essentially means, does it work? You know, does it do what we want it to do? So for example, if I'm developing a drug to cure diabetes, we're then going to be testing, well, does this drug actually cure diabetes? You know, does it do what we want it to do? The second thing we're going to be testing is toxicity. And this essentially means how harmful it is. Okay, yes, my drug may cure diabetes, but if my drug then goes on to grow a second head or something, that's going to be harmful. Okay, so another thing we're going to be testing for is whether it's harmful. And a third thing we need to test is the dosage. And really what we mean by dosage is, first of all, the concentration of the particular drug in the tablet, i.e. how much of the drug or how much of different things should we put within one tablet, and also how often that should be taken. Okay, so not just the how often it's taken, but also the concentration within each tablet. So the two things really about dosage. So in step 1 of our preclinical trials, we're testing on cells and tissues, and we're testing for efficacy, toxicity, and dosage. Now if it were to pass that stage, we would then move on to test our drug on animals. And again in this, we're really testing for efficacy, i.e. does it work? Again we're testing for toxicity, i.e. how harmful it is. And again we're testing for dosage, what concentration should be given and how often that concentration should be given. Now once it's past that, and again many stages do not pass this, many drugs do not pass these stages, it then goes on to the clinical trials. And this is the stages where they really start to test on humans. Okay, now stage 1 of the clinical trials is on healthy volunteers. Now really you're testing for two things in healthy volunteers. They're testing for its toxicity, i.e. is it harmful, and they're testing on the dosage. Okay, now we can't really test on the efficacy with healthy volunteers because again, if I'm testing for diabetes but I'm not testing it on diabetics, then how could we possibly know if it's going to work or not? Because they're not diabetic. So really what we're testing on is it harmful to humans at this stage? And again, really trying to hone in on that dosage. So once it's past that stage, it goes on to a rather small sample of the infected volunteers. Okay, now what we mean by that is, again, let's use this example that I made a diabetic drug for diabetes. We're now getting in a small sample of diabetics, i.e. the people with the disease that I'm trying to cure, to try and test on them. Okay, and here, again, we're testing on toxicity, we're testing the efficacy, and that's quite important at this stage, and also the dosage as well. And again, the final stage, really, once it's gone through all that, we then go to a much larger sample of the infected volunteers. Okay, so stage 2 is a small sample. Stage 3 is a much larger sample of these infected volunteers. And this is going to be a very, very big study at this stage now. And again, testing toxicity, efficacy, and the dosage as well. Now, these stages on humans, are called double-blind trials. And what that means is that half of the participants will receive the drug, whatever drug it is we're testing, and the other half will receive something we call a placebo. Now, a placebo is a substance that's like the drug being tested, but it doesn't do anything. Okay, so the doctor can see the actual difference the drug makes, and it allows for the placebo effect, i.e. when the patient expects the treatment to work, so feels better, even though the treatment isn't actually doing anything. Okay, so in these double-blind trials, the patient doesn't know whether they're receiving the placebo or the actual drug. But the thing that makes it the double blind is that the doctors do not know either whether the patients are receiving the drug or the placebo. They won't get told. So the patient doesn't get told and the doctor doesn't get told. And this is to really reduce that placebo effect. So we're stopping people from thinking they're getting better just because they're taking the drug. Okay. And then once it's passed all these stages, then it can go on to a peer review. Okay, now this is reviewed from scientists in the field to really check the data and check the methodology and to see how it was done. And this really helps prevent false claims, which is again sort of an added layer of protection for us as the consumer, us as the buyer. Before we go and buy a drug, we know it's gone through all of these stages, so we know it should be safe. Okay, so we go preclinical trials, which are on cells and tissues, and then on animals, and then the clinical trials, which is on healthy volunteers, then a small sample of infected volunteers, and then a larger sample of infected volunteers. And these will be double-blind trials, so the patient or the doctor will not know whether they're taking the drug. So what we would now like you to have a go at is answering these questions here, and we'll go through the answers together at the end, so pause the video now. Okay, so let's go through the answers. Number 1, what drug is extracted from foxgloves? Well, that is Digitalis. Number 2, what is Digitalis used to treat? Heart conditions. Number 3, where was aspirin originally extracted from? The bark of a willow tree. Number 4, what scientist discovered penicillin? Well, that was Alexander Fleming. Who discovered penicillin? That's the same question. Question 6. What is the source of most new drugs? Well, that is the rather large pharmaceutical industry. What are new drugs tested for? Efficacy, toxicity, and dosage. What happens in the clinical stages of drug, or the preclinical stages of drug trials? Well, first they're tested on cells and tissues, and then they're tested on animals. What happens in the clinical stages? Well, first they're tested on healthy volunteers, and then sort of unhealthy volunteers, and then a larger sample of the unhealthy volunteers. What is a placebo? A substance that's like the drug. So a substance that's like the drug being tested but doesn't do anything. Number 1, what is a double-blind trial? Well, this is a trial where both the doctor and the patient do not know whether they are receiving a drug or a placebo. And finally, what is a peer review? Well, a peer review is a review from scientists working within the field to test their method and their results to prevent false claims. So we have now there looked at drugs, i.e. how they were originally developed and how they are nowadays developed, and then the stages that drugs have to go through in order to get onto the shelf. If you completely understand this topic, then you can now test yourself with the quiz and answer any exam questions. However, if you do need any help, then please get into contact with one of our tutors who will be happy to help.